Okay, it's about time to start the show. All right, so, yeah, you notice it's snowing out there, and our attendance is really low. That's okay. That's why it's really important I better record all this stuff. About lab this week. So right now in lab, we're just waiting, right? We just got, hopefully you've all got your crosses of the F1s, the hybrids, going, but they're probably not producing flies yet. Many of you still have the F1s to count and score. So I noticed we got something like 80 flies tallied up in the spreadsheet. We can do better than that. Let's get more than that. And then the next step is just count F2s. Now, with the F1s, I told you all, hey, just 50 flies is good enough for each group. That'll make me happy. For the F2s, I want you to just go nuts and count every single fly you can. Yeah, because we're looking for a 9:3 to 3:1 ratio, and those 1/16, those are going to be rare. And so the more flies we count, the more accurate our measure of the frequency will be. So, yes?
So, the most efficient way to do that, would that be like waiting till like there's a bunch in there, or can we like dump them out and then count them?
However works most comfortably with your schedule.
Okay.
Yeah, so you don't want it to go more than about a week or so.
Okay.
So that's your window of time. So you basically say, if I started getting flies, I've got 1 week and I can't do it anymore after that. So, if it's easiest for you just to wait and do it all in one big, massive, eyeball-burning session, that's fine. Or you can dribble it out and do a little bit each time and add them all up at the end. Okay, so that's where we're at. We're just counting right now, and that will probably continue through next week. I might start our next cross next week, but I have to look at the calendar very carefully because there's this annoying thing called spring break. Have you ever noticed? And it's really bad if spring break falls right across the time when I need you to be setting up new crosses. So I try to schedule things so we won't have to worry about that. Because otherwise I have to tell you skip spring break and you have to stay here. That never gets people happy. Anyway, okay. So that's where we're at with lab. We're just counting. I will be in lab the usual scheduled time. So if you just want to ask me general questions about anything, feel free to stop by then. That's a good time. The last homework assignment has been graded, and the answer key is posted. If you're confused by anything on that, just, come ask me. Okay. So where we're at right now is we're starting on chapter 7. I skipped a few. Yeah, I just felt like, hey, I think it's a little easier to get across the concepts of sex determination than it is the mapping stuff that we've got to do next. So let's just look at the determination of sex in genetics. We'll see how that goes. And I looked at what I got planned to present, and it's kind of ambitious. So this is probably not going to be done today or even Wednesday. We'll see. Okay, so here's a simple Mendelian genetics problem for you. So I tell you, the progeny of a cross that expressed a particular phenotype appeared in a 1-to-1 ratio. What would you say was the genotype of the parents and the genotypes of the two kinds of progeny? We have seen this result before with test crosses, you may recall. Yeah, so that would do it. Take a heterozygote, cross it to a homozygous recessive individual, and you will get 50% big A, little a, 50% little a, little a. Exactly what you want. Okay, that's very familiar looking. Because think about this cross. You're all the products of this cross. You have a mother and a father. Two different sexes, and they crossed. And if we did a statistical analysis of all the siblings, of all the people around here, we'd say it's roughly about 50% male, 50% female. So it sure looks like maybe we could say that sex is a simple Mendelian character. That would be elegant. So you just get one sex that's heterozygous, the other sex is homozygous. They come together and you get a 50-50 mix of progeny. It all works out perfectly. It is also completely wrong. That's not how it works. Turns out there's complications to this. Mendel would not be happy with this, the way it actually works. Okay. So, the hypothesis is that one sex is heterozygous for a sex determination trait, and that's going to turn out to be kind of true, and produces two kinds of gametes. So they're called the heterogametic sex. And the other sex is homozygous for the trait, and it's called the homogametic sex. So males in the human species are heterogametic. They can produce sperm that contain X or sperm that contain Y. Y is the male determining trait. And females all produce nothing but X. So sex is specified by the type of sperm that enters the egg. Now, there are going to be complications coming up, but one approach we could take, one approach we can take is, hey, it could be just an entire chromosome that does this job. So there's an X and a Y chromosome, and you have probably heard this many times, that males are all XY and females are all XX. It's not entirely true. It's as usual in this class, everything's going to get more complicated the farther we go. And sometimes sex is not inherited as a Mendelian trait. Okay. One model for this is shown right here. Yes, we're just like bugs. So this is the milk. You've seen milkweed bugs, I'm sure, right? These guys? They're pretty common. There's a lot of different species of these. But yeah, we find these, they kind of crawl around the hallways here even, especially in the fall. And this one is something called a broad-headed bug. I haven't seen one of those. I don't know what they are, but that's what they look like. Anyway, the model here, this is called Ligaeus or xxxy determination. That is, the homogametic sex has a pair of chromosomes, the X chromosomes. The heterogametic sex has one X and one Y chromosome. And in simple versions of the model, the presence of the Y chromosome specifies the sex of the individual. So if you get a Y chromosome at fertilization, sorry, you're stuck. You're going to be a boy. If you get an X chromosome, sorry. Well, two X chromosomes, sorry, you're stuck. You're going to be a girl. Again, it's going to be more complicated than that. That's not entirely true, but okay. Another alternative mechanism is this one, the protonor or xxxo determination. So in this case, the homogametic sex has a pair of X chromosomes. The heterogametic sex has one X chromosome, and that's it. They don't have a homolog to their X chromosome. And there we often see this, that the number of X chromosomes relative to the number of autosomes. Autosomes are the non-sex chromosomes. Specifies the sex of the individual. And that can also be true for Ligaeus sex determination. It's going to get messy. Okay, why? Why do we think this? Well, I gotta mention this person. This is Eddy Stevens. Eddy Stevens was a very interesting person. She worked as a school teacher for many, many years. And then when she turned 39, she decided, I'm going for my PhD. I'm going to become a doctor of biology. I'm going to study cytology. This was a radical thing for somebody to do in the 1900s, as you might guess. For one thing, many schools did not allow women to enroll at all. That was starting to change around this time, so she could enroll. She went to Bryn Mawr in Philadelphia and worked on her PhD there. Specifically, what she studied is, it's all written out here. This is a little article written by Thomas Hunt Morgan, and you'll see much more about Thomas Hunt Morgan later this week, where he points out she found that the male of a beetle produced two kinds of sperm, differing in that one half of the sperms have a large chromosome and the other half a smaller chromosome. So an X and a Y chromosome. And he points out, other people already suggested this. This guy McClung did. But then he pointed out that, Eddy Stevens was the first to, make it really clear, to spell it all out. One of the funny things I want you to notice here, Miss Stevens? This is Dr. Stevens. So it's one of these casual dismissals. They're so annoying. Another thing he points out is he says that Eddy Stevens' key discovery was also shared by this other guy, this man, E.B. Wilson, Edmund Wilson, also a good geneticist, well known as a geneticist. That wasn't true. E.B. Wilson had studied a similar phenomenon, but there was a peculiarity here. He only looked at sperm. He didn't bother to look at ova. Again, there's another interesting distinction that we often see in sciences about how female traits are sort of dismissed. So, no, Wilson does not deserve equal credit with Eddy Stevens. What's really annoying about this too is this is her obituary. So Eddy Stevens struggled for years and years. She got her PhD. She tried to get a position. She was given positions as instructors and things like that, but a full professor? No, you don't do that with women. They don't get to be full professors. She finally got offered a professorship at Bryn Mawr, but it happened to be the year that she died, and she had to turn it down because of ill health. So, this is just another example of a woman getting screwed in science. It's very similar to the case of Rosalind Franklin, who I'm sure you've heard of. Rosalind Franklin made key discoveries in the structure of DNA. She wasn't properly acknowledged. She died of breast cancer before the Nobel Prizes were given out. Same here, she died of breast cancer, which is terrible. But I should also tell you, this story also tells you something else. That much of the lesson here from Eddy Stevens is called the chromosomal basis of the determination of the sexes. That your sex is determined by X and Y. Yet she's an example herself that there are so many other phenomena here. The fact that she was denied a professorship, that she had to struggle so hard to get an education, that even in her obituary she wasn't properly acknowledged for her work. Sex is messy and complicated. Okay. So, Eddy Stevens comes up with this idea. We're going to see it's not entirely true because it turns out there are other mechanisms for defining sex in different organisms. So this is a well-known example, temperature-dependent sex determination. So lots of reptiles don't have X and Y chromosome. The males and the females are genetically identical. But what determines the sex is temperature. temperature in the egg when it's developing. So a great example of those are the turtles we can find around here. I don't know if you're around in the summer, but come the summer you go over by the Pomme de Terre River and you go around there, you'll see turtles constantly coming up out of the water to lay their eggs alongside the road there. It causes some problems. You got to be careful. Don't run over the turtles, please. If you see them there, yeah, stop your car, get out, help them across the road because they're kind of slow. But what they do is they lay a clutch of eggs in a small shallow pit covered with dirt. Eggs are metabolically active. They're producing heat all the time. So you get a temperature gradient across the clutch. So outside, the outermost eggs are kind of cool relative to the innermost ones, which are kept warm. And they use that information to determine whether you develop as male or female. Curiously, in different species, there's different mechanisms. So here you see this is percent female. This is temperature on this axis. In some species of reptiles, when you cool them down, they make all females. In other species, when you heat them up, you make all females. And yet other species, they show this kind of distribution where the extremes are females, either extremely warm or extremely cold, and it's that narrow middle in between that develops as males. And this works out well enough for turtles. Okay, so that's an alternative mechanism. And it should tell you right away, there's got to be something more going on genetically than just a single chromosome. But there must be a whole bunch of interactions that take place. This is one I'm not going to get into. I'm not going to test you over this, because this is so confusing. There are species that have more than one sex chromosome. So this is just an example of a frog. This frog has 6 sex chromosomes. It doesn't just have X and Y. It has this arrangement you see up here. There's the female, these 3 pairs of chromosomes, and the male has these other arrangements with a very complicated breakup of these patterns. This happens sporadically in other species. The notorious one is the platypus, which has 10 or a dozen different sex chromosomes. And I don't know how that works. Yeah, you know, if any of you want a senior seminar that'll be very, very challenging and fun, try to explain these other mechanisms of sex determination. Here in this frog, you can see down here, they've got a little hint of what's going on, is that they form these complexes of multiple chromosomes during meiosis that then segregate appropriately. But, oh man, this is a mess. So yeah, I'm waiting for one of you to explain it to me someday. You'll do that, right? This would be a hard one for a senior seminar, but yeah, it'd be fun. Okay, here's the way we do it in humans, though. This is a human karyotype. There's X and there's Y. Great big X and a little bitty Y. And the correlation seems to be pretty strong. That is, if in every genetics class I took blood samples from each of these students and looked at your chromosomes, I'd have almost perfect accuracy in predicting whether you're male or female. Almost perfect. This is, by the way, one of the reasons we don't do that in the genetics lab, because you don't want to be told you're not the sex you think you are in the middle of a genetics lab. No, seriously, way back when I was a graduate teaching assistant, we had a situation like that. Oh man, that was horrifying. This poor woman just does a routine karyotype of herself, and surprisingly noticed, oh, I've got a Y chromosome. You don't want to find that out in the middle of a genetics lab, okay? So yeah, we don't do that. We also don't look for bar bodies, which would be really easy. And we'll talk about those in a moment. So anyway, there are these different arrangements of these chromosomes that occur. Usually XX means you're female, XY means you're male, but not always. Another issue here, heterogamy. So, I told you, okay, we got a heterogamic sex. You may have noticed when I talked about those, I didn't say male and female. That's because it's not always a close correlation that way. So in humans, yes, males tend to be the heterogamic sex. That's also true in insects, like our fruit flies. But in butterflies, birds, and some anemones, the female is the heterogamic sex. Okay. And just to avoid the confusing nomenclature, in those species we say they're ZZ, ZW instead of XXXY. But it's still in principle the same thing. Okay, so chickens have 78 chromosomes. And the female chickens have 76 plus a Z and a W chromosome. Whereas the roosters all have 76 autosomes plus 2 Z chromosomes. Already we're starting to see that sex may be getting a little more complicated than Eddy Stevens might have thought. So let's look at these chromosomes. Let's look at the Y chromosome for a minute. So there's the general structure of the human Y chromosome. The human Y chromosome is mostly empty junk. Lots of repetitive DNA. All that repetitive DNA makes it challenging to sequence them. So it seems like every year they slightly modify the number of protein coding genes they found in it. They found another one. They found one, oh, that wasn't a protein coding gene, that was a mistake. That varies a bit. But roughly 55 protein coding genes, that's it, on the Y chromosome. Put that in context, remember you got 20,000 genes in your whole genome. So the Y chromosome is surprisingly runty, with very little on it. What do you find on it though? Well, there's a couple things. There are pseudo-autosomal regions. What are these? These are, remember when we talked about meiosis and how you had to get the homologous pairs lining up? The X and the Y chromosomes are the homologous pairs, even if they have very different structure. And the way they find each other and line up is because they have these key regions, the pseudo-autosomal regions, that are the same between an X and a Y. Everything else may be different, but these pars allow them to pair up during meiosis. There's also a large chunk of it that contains non-recombinant regions of the Y. That is most of the chromosome. They're called non-recombinant because remember that there's this huge size difference between X and Y. You don't want to have crossing over. Crossing over messes things up a lot if it happens between the sex chromosomes. And we'll talk more about that when we go back to gene mapping and fun things like that. So it's mostly junk, but there are a few things. There are some X homologous genes. What does that mean? That means these genes are also present on the X chromosome. So some, we maintain a pair of genes that are just on an X and a Y. There are also some genes that are testis specific. Testes are, well, and ovaries too. Everything is complicated. But testes have a lot of complicated things they have to do. That sperm, for instance, have to mature inside the testis. They go through meiosis, but then you have to do all the stuff like strip out all the cytoplasm, stick a tail on there, a flagellum. You have to shape them to be sperm. And that takes months. So they're sitting there for months and constantly being modified. You also have to add specific molecules on the surface. so that they are recognized by an egg at some point. Yeah, you can't bring together, for instance, dog sperm and human ova, because the human ovum will reject them because they don't have the right stuff on their outside, the wrong carbohydrates, the wrong proteins. So they have a very specific set of things that have to be done there. So that's all done in the testis, and there's a bunch of genes on the Y chromosome that play a role in that kind of pattern of marking the sperm. And then the region we're going to talk about here, sex-determining region Y. It's right up there. It's a little tiny thing. Sex-determining region Y is the one that is responsible for triggering male development in the embryo. Okay. Now, it's really easy to take a very simple approach to this and just say, okay, SRY, if you get SRY, you're male. But sex differentiation is a very complex process, as you see in this little diagram I have over here. But TDF contains this thing, or SRY contains this protein called TDF, testis determining factor, and it plays a very important role in specifying the sex of the individual. And we know this because there's a whole bunch of experiments that have been done. So there are cases where we have XY individuals. You look at their karyotype, and they got an X and they got a Y. But there has been an accident, and we've deleted TDF or SRY from that Y chromosome. And what happens to those individuals? They develop as female. Okay, that's a good clue. This is important. We also have cases of XX individuals. Again, you look at their karyotype, you say, oh, XX, you're a female. But they also have a copy of SRY that has been moved to another chromosome. And that SRY then induces them to develop as male. See, this is the kind of thing I'm talking about when I say we don't do these tests in your undergraduate genetics labs, because you might get that surprise that you can have individuals who fully identify as male, who have all the physical characteristics of a male, and they're XX. And vice versa. People who identify fully as female, and they got a Y chromosome. There are other subtle ways that these changes can occur too. Also, we can do the experiments. Yeah, you can just get in there and you can make transgenic mice where you stick SRY wherever you want. And if they've got an SRY, they develop as male. Okay, now, I want to emphasize though, it's not always that simple. See this hairy diagram over here? There's SRY. SRY, among the things, it will do many things, but among the things it will do is induce this transcription factor called SOX9, which then activates a whole bunch of things. It is dependent, for instance, on this other molecule, FGF9. If you have if you delete FGF9, SOX9 won't be activated. You won't get a test. Yeah, it's, I've shown you epistasis. This is epistasis. It's really complicated. All kinds of mutual interactions in here that are necessary to produce the testis. So there are opportunities for mutations in all of these named proteins that can lead to changes in sexual differentiation. Okay. What about the X chromosome? X chromosome is huge. X chromosome is the 8th largest chromosome. It has about 900 protein coding genes. And over here, I just threw in a short segment of the gene map for the X chromosome. It's unreadable. Don't try. It's too hard to read. There's so much stuff there. There's all kinds of things packed onto the X chromosome. You may recall there's an opsin gene on here, a gene that is responsible for assisting with color vision. It's on the X chromosome. The X chromosome is not just the girly chromosome, okay? Having an X chromosome isn't just for females. Males require this. There are even some testis-dependent genes on the X chromosome. There are also testis-dependent genes on autosomes. It's all a tangled mess in here. Okay, so some of these genes are essential for male development. You can have mutations that knock out a gene on the X chromosome, and in an XY individual, they induce variations in normal sexual development. Oh, wow. So, really, it can get very confusing here. For example, one of those genes is a gene called DAX, DAX1. What DAX1 does is it suppresses the function of SRY. So you get SRY yelling at the cell, you're going to make male structures, express as a male, make male hormones. But at the same time, there's a gene on the X chromosome, DAX, which is saying, no, shut up, SRY. Now, in ordinary males, you got one copy of DAX, because you only got one X chromosome, you got one copy of SRY, and it's like DAX is just kind of quietly suggesting, no, stop being so male, okay? And the SRY can just ignore it, and you go ahead and develop as male. In some cases, though, there are individuals who have a double dose of DAX. They've got two copies of DAX on their X chromosome. And what that leads to is feminization, because now those two DAX genes can team up and shout loud enough to suppress some of the functions of SRY. Yeah. Now, it gets more complicated than that, though, because DAX, it turns out, even guys want one copy of it because it assists in promoting this hormone over here, AMH, which is an andronizing hormone. It is responsible for proper differentiation of the tubes and pipes in your reproductive system. So again, if you haven't taken embryology, you won't know this, but males have a set of Wolffian ducts, females have a set of Mullerian ducts. Early in the embryo, they have both. And one of the things AMH does is it tells your fetal boy, get rid of your Mullerian ducts. So it's subtle. There are males walking around who have both sets of ducts. So you want this turned on if you're going to make a male. But this depends on DAX1 as well as SRY, SOX9, etc. Again, epistasis. I will say that many times in this class. There's lots of interactions that have to go on to produce these gene products. Sexual differentiation in general. Now, this is unreadable because it's so tiny, isn't it? If anybody wants, I can send you a full-scale copy of this. What this is a diagram of all the gene interactions that are necessary to differentiate into male and female. And it's spaghetti, isn't it? There are all these tangled interactions that are going on in order to get that differentiation. So we're going from conception to puberty here. There's a whole bunch of interactions that have to go on. I mean, seriously, you probably remember this. You remember puberty? What a mess that was. You are going from a small child who's got some sexual characteristics, and then you go through puberty, and all of a sudden you get a whole bunch of new ones, secondary sexual characteristics added, maturation of the reproductive system. and all kinds of brain changes that go on. So there is this constant refinement of sexual differentiation that goes on from birth actually all the way through to death. So this is just to tell you, yeah, it's complicated and messy. If they ever let me offer my developmental biology course on this, we could spend a month on just this phenomenon. Another factor I want to point out, and this is kind of current, because this is from an article written by, there you go, this well-known embryologist named Keith Moore, who in 1968 was getting really upset by the Olympics. Because in 1968 they were doing all this testing. They were pulling aside athletes and giving them these very invasive tests to say, are you really a girl? Are you really a boy? Do you have an XX chromosome set or an XY chromosome set? This still goes on nowadays, of course. We have lots of different ways that we assess the so-called biological sex of an individual. But Keith Moore is saying, no, you can't do that. That doesn't work. Because sex is not simply chromosomal. There's a heck of a lot more to it than that. So Moore focuses on the 9 components of sexual phenotype. He says, no, it's not just one thing. It's not just whether you have a particular chromosome or not. There are 9, count them, different phenomena that are important in defining sex. So he lists them here. So external genital appearance. You may say, hey, that's enough. Do you have a penis or do you have a vulva? That's all it takes to determine whether you're a boy or a girl, right? But then he points out, but then there's also internal reproductive organs. There are women walking around with a normal external anatomy, but they don't have ovaries. They're still women, right? Same with males. There are males walking around who may have abnormal or missing testes. They got everything else. So, internal reproductive organs, that's kind of important. Then there's also the structure of the gonads. Testes and ovaries, if you've ever looked at a cross-section through them, are not simply potato-like structures. They have all kinds of differentiated cells in them that play various roles in sexual behavior, maintaining sexual appearance, and all this kind of stuff. So they're all scattered in there. And there are individuals who exhibit a syndrome where their ovaries, for instance, may have patches of testicular tissue within them. Are they boys or are they girls? Just on that one criterion, you'd have to say they're both. Then there's one you're probably familiar with, endocrinological sex. What hormones do you make? Boys all make testosterone, you say. Girls all make estrogen. When you actually look at individuals, though, you find, all you women out there, you make trace amounts of testosterone, as well as estrogen. And it turns out that testosterone is really important for sexual responsiveness. So you're making both, just a lot more estrogen than testosterone. And males as well are making testosterone and small amounts of estrogen. And the amounts of testosterone vary. I hate to admit this, but as you get older, your testosterone levels drop. And it doesn't change your sexual behavior at all. In fact, there's really poor correlations between testosterone levels and sexual interest, sexual behavior, also between testosterone levels and athletic performance. So he's making that case there. There's genetic sex. Do you have SRY or not? There's nuclear sex. Do you have a bar body or not? Chromosomal sex. Do you have an X chromosome or a Y chromosome in there? Then also psychological sex. This is real. what do you think your sex is? How do you feel up here about sexual activities? And then also social sex. Where do you fit in society? Where does society recognize you? What role are you trying to conform to? So Keith Moore lists all these things, and he's pointing out you can have an individual who's clearly male at one of these, and then female at another one, male at another one, that you are all a mix of these different types. And so what he was arguing is you can't just do those cheek swabs and look at the chromosomes. It's not an adequate way to define sexual behavior, sexual or true sex, as people like to call it. So it's all messy. I think that's the main lesson here. There's some general properties that go along with sexual determination, but it's always much messier than you might imagine. Okay, I mentioned bar bodies. Here's an example of a bar body. So there's a problem in this kind of sex determination. Okay, women have 2 X chromosomes, men only have 1, and then they have this pathetic little Y chromosome that doesn't have much in the way of genes on it. That is a dosage problem. Because that means that women could have twice as much of every gene product on the X chromosome as men. Now, we'll be talking later about some of these disorders that occur with imbalance in chromosomes. That, for instance, Down syndrome is caused by just having an extra chromosome 21. Yeah, we all know Down syndrome is pretty obvious. So, how do we cope with the fact that all you women have 2 X chromosomes, whereas your professor standing up here only has 1? I should be sick. Right? Maybe I am, but we males should all be weaker in some sense. Well, the way we have gotten around this biologically is that females all inactivate one of their X chromosomes. So you've got 2 X chromosomes, but you shut one down. So you tie one arm behind your back so that you can compete fairly with males. Actually, it's more substantial than that, because sometimes excessive gene doses can cause all kinds of problems. So it's a good thing that you do this. When it is inactivated, it forms this thing called the bar body. So the bar body is a visible particle. These are some photos of some cells. There's a male cell, set of male cells, set of female cells, and all the female cells have this little dark body scrunched up against the nuclear membrane. That's called the bar body. Now, if you watch crime shows, you know those pathologists, you dig up some fragment of a body, some murder victim somewhere, all you got is their left hand or something. You can pull out your trusty microscope and you look at the cells there and you can determine whether it's from a male or a female by looking for the bar body. So this works pretty well in lots of cases. Okay, so we got this really obvious distinction. There's an interesting phenomenon associated with this. This is Mary Lyon, and she came up with something called the Lyon hypothesis, which is about which X chromosome gets shut down. You got 2 of them. So which one is it? And she proposed a couple of things. One is that it's random which one gets shut down. So yeah, you got 2 X chromosomes, you're just going to turn off one of them in each of your cells, and it's entirely random which one it is. The other thing is the choice is made early in development. So it's not like the instant the zygote was fertilized, it picks which chromosome to shut down. No, it goes through a bunch of rounds of division. The embryo may get up to 1,000 or more cells. And then all of the cells independently choose which chromosome to shut down. So that's random, and then it's made early, and you have a mix of cells. Some turn off one X chromosome, others turn off the other one. And once that choice is made, all the progeny of that cell keep the same X chromosome inactivated. That is, they go through mitosis, they temporarily unravel the bar body, duplicate it, and then the same chromosome is shut down in both of the progeny. What this means is that you have a clone, the descendants of all the cells that are turned off, for instance, the paternal X chromosome, and another clone that's got all the descendants of a cell that turned off the maternal X chromosome. And you can get this kind of patchy appearance. This is how we explain calico cats. Calico cats are all female. Well, almost all female. There are some special cases where they're not. But they're female. They have a gene responsible for pigment color that's located on the X chromosome. So one chromosome has a gene for red fur color, the other chromosome has a gene for X fur color, and depending on which one gets shut down in a set of cells, you either get reddish fur or black fur, and it's random. So calico cats all have this random assortment of patchy colors on the surface of their body. You might be saying, well, what about this white fur, like the belly fur and all that? That's a different gene. So there's a different gene that's regulating the color here. So it gets complicated. But anyway, Mary Lyon is the person who came up with this hypothesis. It seems to work out pretty well. It's been tested thoroughly, and it seems to be valid. Now, you might ask, where are the calico women? Well, we have different arrangements of genes, so human women do not have skin pigment genes on their X chromosome. It's such a missed opportunity. It would be so cool, wouldn't it? We do have some examples like this one. This is not entirely a good thing. Yeah, well, I'd say it's not a good thing at all. There is a disease called ectodermal dysplasia, a genetic disease, which makes your sweat glands fail to form. So you find these individuals who have these patches all over their body where they're not making any sweat glands, which means the skin there is rough, and you've got to moisturize all the time and things like that. But it's these distinct mosaic patches. So yeah, if this were a fur color gene on humans, you'd see these kind of patterns on them. But unfortunately, it is an awkward and unpleasant disease, which causes discomfort to the poor women who have it. Obviously, of course, if men have it, well, men either don't have it or they have it all over their bodies. So that's even worse. Also, red-green color blindness. We mentioned you got this opsin gene for color in your eyes. It's also kind of a patchwork in your eyeballs. Which one gets turned off? So if you're a carrier for color blindness, some of your retinal cells are colorblind. Just the way it works, because those cells inherited an X chromosome in which the defective opsin was expressed. But because of the way our vision works, you don't notice. All of your vision is assembled by your eyes are constantly moving and sweeping across the field of vision, and they're sending information back to the brain, and the brain then reconstructs that to get color, so you don't see it. Okay. Now, what about Drosophila? Yes, that's what we really care about, this human stuff. Who cares? What about Drosophila? Drosophila also has that XXXY pattern of sex determination. So in some ways, they're kind of like us. Yeah, you can look in a Drosophila chromosome or Drosophila cell and count chromosomes, and you can see, hey, they got an X and they got a Y. If they got two X's, they're female. If they got an X and a Y, they're male. But there are some major differences. So just to suggest that there's some subtleties in how this pattern works out. So there's three major differences. Here's one. In fruit flies, the Y chromosome doesn't define sex. It's kind of a boring placeholder. It does nothing for sex determination. Instead, what it is sex is specified by the ratio of X chromosomes to autosomes. All that matters is how many X chromosomes you have relative to the number of autosomes. So here's an example of all this. So your typical female fruit fly is right there. She's got two sets of autosomes, 2A, and she's got two X chromosomes. The ratio of X chromosomes to autosome sets is 1.0. So you're female. If you've got a male fruit fly, let's see, where is he? Here, down here. The male fruit fly has two sets of autosomes. He's diploid, remember. He's got an X and a Y chromosome. The Y chromosome doesn't count, it doesn't do anything here. So he's got a ratio of X chromosomes to autosomes of 0.5. And that makes him male. Now with fruit flies, you can play all kinds of games. Yeah, there are ways to induce non-disjunction, for instance, in dividing cells, and partial non-disjunction, so you only get certain chromosomes duplicated. So you can do fun things like experiments like here. Here's a tetraploid fly. He's got 4 sets of autosomes, and he's got 3 X chromosomes. 3 X chromosomes here. That means the ratio is 0.75, which is halfway between 0.5 and 1.0. So this is an intersex fly. So that's all it takes to make those. You can also get this case up here. So normal diploid, we've got 2 sets of autosomes, but now we've made an extra X chromosome. There's 3 X chromosomes here. This is now a ratio of 1.5. This is called a metafemale. It used to be that these were called super females, which is problematic because it turns out there's a similar condition in humans, and you don't want to go around labeling certain individuals as super females. It's also problematic to often call people intersex. So anyway, you got this range, and so it's really just the ratio that specifies it. It's not like there's an SRY in flies. They're just titrating out the quantity of products that we see. Here's another difference, the dosage compensation. So I told you, all right, human women shut down one of their X chromosomes so that they've got the same dosage as human males. Flies do it differently. So then what you do is you crank up the amount of RNA produced by the chromosome in the male. So male X chromosomes work twice as hard to produce the same amount of RNA as X chromosomes in the female. It's still dosage compensation. It's just different. Instead of handicapping one sex, you're taking the other sex and making them work twice as hard in order to produce the same quantity of RNA. Okay, then another big difference is this one. So sex in flies is specified on a cell-by-cell basis. What does that mean? So, for instance, if we look in our tissues, we would find you girls, you women, you're typically XX, and here I am, I'm XY. We could look at my cells, and there will be occasions in my tissues where I am missing a Y chromosome. This happens with increasing frequency as you get older. So I'm just plain X, I'm XO in some of my cells. Hypothetically, I haven't checked. But they're still male. Why are they still male? Because in humans, maleness is often the product of hormonal sex. So hormones are washed throughout your body. So I am loaded with testosterone. So my cells have no choice but to be masculine or male. If you're missing testosterone, if you've got lots of estrogen, your cells, no matter what their chromosomal content, will differentiate as female type cells. That's not true in flies. So here's an example, just a cartoon. So this fly lost one of its two X chromosomes. So it started out as female. But it lost one of them by an accident during early mitosis. So half of its cells are XO and half are XX. And because of the way flies develop, you can even see nice tidy seams here. So this side, for instance, is the female side. It's got two X chromosomes. And so that will obscure recessive traits. White eyes, for instance, is a recessive trait. So you won't see it here. This fly is, in all of these cells, it's XX, so it's got X white eye color, X red eye color. And red is dominant, so it'll be red. On the other side, it's lost one of those X chromosomes. So now it will express the white phenotype. And we can combine multiple mutations in these flies. So this is a fly that's got a white eye mutation, and it's also got a short wing mutation, miniature wing. It can have others. I don't see any expressed here. But in your next experimental cross, you're going to be working with flies that are triple mutants, white, miniature, and forked, funny bristles. We'll talk more about that when we start it. It would be kind of cool if when you're doing this experiment, you look very carefully at your flies, because it's possible, it's rare, but it's possible we'll get some gynandromorphs out of it. Haven't seen them in years of doing this, but it's still a possibility, so keep an eye open for that. Okay, so this is a case of mosaicism. And in flies, since it's cell by cell, you don't have any choice. If you're a cell with the XX chromosome, you are going to develop female structures. So, for instance, you can even look at the genitals of these flies. And they're half male, half female. They will make a testis on one side and an ovary on the other. All kinds of fun stuff like that. In humans, sexual differentiation is not cell autonomous. Your cells depend on signals from hormones in particular to determine how they differentiate. Now, like I said, spontaneous loss of chromosomes does occur. This was a surprising result. They studied blood donations. They just went through all the blood donations that were made at some hospitals, and they looked at the white blood cells and karyotyped them and asked what was going on. And they found that in men over 70, roughly half of the men, a little less than half of the men over 70, have blood cells that are type XO. They've lost a Y chromosome, which is really kind of interesting. In addition, the same thing happens in females. So there are women whose blood, I haven't seen the quantities exactly, whose blood cells are XO, which is still going to develop as female, but they've got a difference in their chromosomes, which is kind of fascinating. Also, another thing that happens in women is mosaicism in women who have children. that, yeah, if you grow a baby in your womb, and then you give birth, I mentioned before, it's splatter city, there's blood and fluids flying all over, and populations of your offspring's cells will find their way into your tissues, particularly as stem cells, and you can find them all over the place. So they dissected some poor 94-year-old woman who had died and given her body to science, and they looked in her brain, and they found little clusters of cells that were male, that were derived from her offspring. Now, is that creepy or what? Yeah, you're never going to have children now, right? Because it's like an invasion of the body snatchers here, where you're going to get populated with these things. Again, it's small numbers of cells. It doesn't turn you into your children. Yeah, it's just a really curious biological phenomenon. Okay, another factor here is I started off by telling you about sex ratios and how, hey, it looks like sex could be a simple Mendelian trait. It's not, because there's all these other factors that can influence it. So here's a couple of things that occur. X and Y bearing sperm are not necessarily made in equal numbers. There are individuals who make exclusively Y-bearing sperm and others who make exclusively X-bearing sperm. Another factor is that these two kinds of sperm don't necessarily have equal mobility and viability. It's a general fact that sperm carrying Y, because Y is a small chromosome, are slightly, just slightly faster than X-bearing sperm. Makes sense, right? They got less of a load. Another observation that's been made is that if you have a Y-bearing sperm, it's going to have less endurance. It's going to die off sooner than the ones bearing X-bearing sperm. So there's little differences like that. So differences in mobility and viability. And another important one, this one was ignored for decades because, hey, the egg, it's passive, right? It will just take whatever the male delivers. That turns out not to be true. But eggs have all kinds of receptors and proteins on their surface, and eggs can be choosy too. They can reject sperm that comes at them if it's got attributes they don't like, things on their surface they don't like. And there have been cases where couples discover that they can't have children, and the reason is that the woman's egg rejects male sperm from that individual. So she could presumably have children with other men, but not with the one she chose to marry. Yeah, it gets complicated. These are just some illustrations. There is a sperm bank in St. Cloud. If you're ever interested, you should call them and see if you can do a tour. They can show you all the rooms where they collect sperm. They will also show you how they grade and store sperm. I've been there to look over their facilities. It's actually kind of neat. You know, they pull up sperm samples, they get them on the microscope, and they grade them. ABC, you see here. Grade A sperm are very motile. They go right to their destination. They're very active. Grade B, they are kinked. They tend to go in circles, which is not a very effective thing if you want to travel up a fallopian tube. So they're all messed up. And grade C, they just sit there looking dead. Maybe a little twitch now and then. And you can evaluate men's sexual ability, their ability to procreate, by looking at their sperm and seeing these kinds of differences in them. Also, you sometimes find really weird things like sperm with two heads, sperm with two tails, sperm with kinked tails. Yeah, it's very informative. Okay, and we're at just about the end. So I will stop here. We'll finish this up on Wednesday. I want to talk about the sex ratios and a few other things. And then we're going to move on to discussing sex-linked traits and how they get inherited. And this will add new complications to the kinds of genetics problems I can throw at you. Okay, go be safe. Watch out on those roads. Watch out on those sidewalks. All those things. We'll talk to you on Wednesday.